Hi everyone, welcome to Bio 35. My name is Tran and I will be helping you learn about the five senses today. Our sensory system serves to take information from our environment and convert it into neural signals that we can react and behave to our environment correctly. In other words, our senses allow us not only to survive, but to interact, enjoy, and understand the world around us. The five senses are vision, hearing, taste, smell, and touch. So what is on the agenda for today? First, we will define what sensory transduction is and what it looks like in each of the five senses. Second, we will break down how each specific sense processes stimuli from the environment and our sensory system. Lastly, we will compare the similarities and differences between the senses as well as their processes and have an overview of what we learned. So let's get started. So first, what exactly is sensory transduction? Sensory transduction is when our body converts various forms of energy located outside the body into neural signals or action potentials. How do we do this? Well, we have different types of receptors. The specific form of energy that receptors convert depends on which sense we are talking about. Take vision, for example. Sensory transduction vision refers to how we convert light rays, specifically photons, in our environment into neural signals using photoreceptors. When hearing, that energy comes in forms of sound waves with hair cells. Similarly, when tasting or smelling, chemicals are detected and processed by chemoreceptors. Lastly, for touch, energy comes from mechanical forces and are processed by multiple receptors detecting heat, pain, light touch, and other things. Although each sense differs on what form of energy they receive and what receptors are activated as a result, it is important to understand that all senses have the same end goal, to convert whatever form of energy they receive into neural signals for our body to process. So let's take a look at all these senses individually. Vision, as I mentioned before, utilizes photoreceptors to convert photons into neural signals or action potentials. Here are some key players within the visual system. First, we have two types of photoreceptors, or neurons, on the retina, rods and cones. Rods are responsible for lights and shades, whereas cones are responsible for detecting colors. Next, inside the rods, there's a G-coupled protein receptor called rhodopsin. Inside the rhodopsin, there's retinol, which is light sensitive. Lastly, we have our G-protein transducin, which causes a protein cascade when activated. When light is taken in by the eye and hits the retinol undergoes a conformational change, meaning its molecular structure turns from cis to trans, therefore also changing the structure of the rhodopsin it is found in. Then, when the G-coupled protein receptor rhodopsin undergoes a conformational change, it activates its G-protein transducin, which causes a protein cascade, and a downstream effect of hyperpolarization. What is especially important about this protein cascade is that it amplifies the effects of a single proton, therefore increasing our sensitivity to light. This is how structure and forms function in the context of vision. So now that we know about the mechanicals, let's briefly go over how our visual field is represented in the retina. Let's allow blue to represent the left side of the visual field and red to represent the right. If we trace the left side of the visual field first, we can see that the left field of vision is taken on the right sides of each retina, which makes its way down to the optic chiasm, down the right optic tract, and into the right visual cortex. The right visual field does the same thing but with the left tracts. Vision has retinotopic maps, which mean locations next to each other on the retina are usually represented by locations that are adjacent to each other when we look at things. Next, let's talk about the auditory system and how it converts sound waves, especially pressure waves, into neurosignals. However, unlike vision, hearing depends on another type of receptor, mechanoreceptors, located on hair cells to detect movement from sound waves. Hair cells in the ear are found on the cochlea and cause action potentials, excitation, or stop activation, inhibition, depending on what direction the sound waves push its mechanoreceptors to. Like vision, hearing also exhibits the concept of structure and forming function. The base of the cochlea is tight and thick and is therefore allowed to detect higher frequencies and sound waves. On the contrary, the apex of the cochlea is much thinner and floppier and therefore detects lower frequencies. The hair cells on the cochlea are organized into a tonotopic map, which means that they are organized in a gradient where hair cells next to each other on the cochlea detect similar frequencies because of the structure of the cochlea itself. Much like vision, the auditory system sends its signals to a cortex, the auditory cortex, to process sensory signals. The auditory cortex is also organized by topography and corresponds with parts of the cochlea. The left side of the auditory cortex corresponds to the apex of the cochlea, processing lower frequencies, while the right side corresponds to the base of the cochlea, processing higher frequencies. The next two senses we'll cover go hand in hand, taste and smells. Taste and smells both convert chemicals from the things we eat or the air we breathe to action potentials through the help of chemoreceptors. Both systems rely on the activation of multiple receptors, but there are differences between how these two senses work. In taste or the gustatory system, there are 5 different tastes we can have: bitter, salt, sweet, sour, and umami. The taste buds, unlike the previous systems of auditory and vision, have no discrete expression patterns. In other words, they are random and have no gradient organization. However, there are some similarities to how the visual system is organized. For example, the gustatory system also has a G-coupled protein receptor and G proteins, which cause a protein cascade. However, the gustatory system adds a step, the trip channel, which we will discuss in a little bit. The taste receptors, or the G-coupled protein receptors, are heterodimers, meaning that 2 G-coupled protein receptors must come together in order for us to taste flavors, depending on which 2 receptors are put together. When these G-coupled protein receptors dimerize, it activates their G protein, gusteucin, causing a protein cascade. These TRP channels, called TRPM5 or TRPM4, open and cause a short depolarization, a positive potential, ultimately processed in the gustatory cortex. Now, moving on to smell or the olfactory system, specific odors trigger a particular set of receptors. These receptors are utilized in different combinations depending on which scent is detected. Some only activate one, some receptors activate the same receptor, while some activate multiple receptors. Much like the gustatory system, multiple receptors in the olfactory system can be activated at once and are put into different combinations to react to different stimuli. The olfactory system is more similar to the visual and auditory systems than the gustatory system is, because the odor structure determines which receptors it will trigger and its function. However, odors are not expressed linearly like the auditory and visual cortex. Instead, the olfactory system has a chemotomic map, which means that structurally similar odorants stimulate overlapping but distinct domains in the olfactory bulb. As an example, this part of the olfactory bulb will be activated when you smell body odor, whereas this part of the olfactory bulb will activate when you smell citrus. Lastly, we have touch. Touch detects a stimuli of a variety of things, such as pressure, vibration, itch, finer details, etc., into neural signals through the help of touch receptors. These touch receptors are similar to the gustatory and olfactory system because different receptors are activated depending on different types of stimuli detected. These touch receptors, due to their location on the skin, are responsible for different stimuli. For example, if the receptors are closer to the surface of the skin, it is responsible for smaller areas and has more tactile acuity, therefore detecting finer details and edges. If the receptor lays deeper in the dermis, it is responsible for larger areas and has less tactile acuity, meaning it would detect less sensitive stimuli. After receptors are activated, they are transferred to the homunculus in the somatosensory cortex in the brain via sensory fibers, such as the slower C fibers for duller pain and fast A delta fibers for sharp pain. The homunculus, like the visual and auditory system, an example of topographic organization, where spatially adjacent stimuli on sensory receptor surfaces are represented in adjacent areas of the homunculus. However, the representation of these body parts and receptors are proportional to their tactile acuity. The higher the tactile acuity, the more largely it is represented in the homunculus. Now let's see if we can draw some similarities and differences when comparing all of the systems. So, going down the list, let's first compare stimuli and forms of energy. For vision, they take forms of energy in photons and light rays. For hearing, we have sound waves and pressure waves. Taste is chemicals in the food that we eat. For smell, it's chemicals in the air that we breathe. And touch, it's pressure, vibrations, light touch, and itch, etc. For receptors, vision uses photoreceptors. For hearing, we use hair cells or mechanoreceptors on the hair cells. For taste, there's multiple chemoreceptors. For smell, it is the same thing, but odoreceptors. And for touch, it's touch receptors and differs depending on the location. For topography, vision has a retinotopic map, which means that locations next to each other on the retina are usually represented by locations that are adjacent to each other when we look at things. For hearing, we have a tonotopic map, in which hair cells detecting similar frequencies are located adjacent to each other on the cochlea. And for taste, we do not have a topographic organization because it is random and not linear. The taste buds are not organized in any specific pattern. For smell, we have a chemotopic map, but is not linear. This is where structurally similar odorants stimulate overlapping but distinct domains in the olfactory bulb. For touch, we have homunculus, which is adjacent areas of the body are represented in adjacent parts of the homunculus. Not only this, but the higher the tactile acuity of the body part, the larger representation it has in the homunculus. Moving on, let's see which senses have G-coupled protein receptors. For vision, we have rhodopsin. For hearing, we do not have one. For taste, we have T1R1, T1R2, T1R3, and T2R, which remember dimerize and come together to taste specific tastes. For smell, we have none. For touch, we have none. Next, for G-protein cascades, for vision, we do have a G-protein called transducin, and yes, it has a protein cascade, which allows for more light sensitivity to single photon. For hearing, we do not. Taste, we have gusteucin as our G-protein, and yes, there's also a protein cascade. For smell, there's none, and touch, there's none. And the extra steps, we have vision. So vision travels through the optic chiasm, the optic tract, the visual cortex, and the images remain inverted. For hearing, there are no extra steps. For taste, remember that the TRP channels are added on to at the end of the protein cascade and includes TRPM5 and TRPM4. The activation of a TRP channel causes short depolarization, aka an action potential, and completes the pathway. For smell, we have none, and for touch, we have sensory fibers, mostly C fibers and A delta fibers, which carry signals from the peripheral nervous system to the CNS. For the processing centers, and for vision, we have visual cortex. For hearing, we have the auditory cortex, which is organized with topography and corresponds to the parts of the cochlea and processing frequencies. For taste, we have the gustatory cortex. For smell, we have the olfactory bulb. And for touch, we have the somatosensory cortex, but more specifically, the homunculus within the cortex. Thank you so much for listening, and I hope that you enjoyed my project.